Hello and welcome. I'm your host, Neil Howard. Thank you so much for joining us for another segment of Health Professional Radio. We're going to be talking with Ethan sa-mRNA. He's joining us here as Vice President, Research, CSL R&D Seqirus Vaccines Innovation Unit, and he's also a study author. He's joining us to talk about some key findings from some data that was published in Molecular Therapy, Methods and Clinical Development. And he's also going to talk about the company's next technology platform. Welcome to Health Professional Radio, Ethan sa-mRNA. Thank you for joining us this morning.
Thank you very much for having me.
Well, tell us a bit about yourself and your role at Seqirus.
Sure. As you mentioned, my name is Ethan sa-mRNA. I'm the Vice President of Research at CSL R&D and the Seqirus Vaccines Innovation Unit. I lead the research team in understanding infectious diseases and developing vaccines against them that can offer the highest efficacy. As you may know, we have a specific focus on influenza at CSL Seqirus, as we are the second largest flu vaccine manufacturer worldwide. However, we have a really broad view to advance protection against vaccine-preventable diseases. And on our team, particularly in research, we focus on a variety of technologies to really drive the advancement for vaccines, including established technologies, new vaccine technologies, and ones that I'm going to talk about today is this new sa-mRNA vaccine technology, which we'll get into. Just as a final note, I'm actually very glad that we have this conversation at this particular time. As you all may know, the influenza season started early, and the rates are quite high, whereas there are lower vaccination rates. So we certainly support the U.S. recommendation that people who are eligible should get vaccinated.
Well, first of all, why are we hearing almost as much about the flu as we were hearing about COVID? Do you expect a pandemic almost like COVID, but with the flu instead? And the second question is, what is sa-mRNA?
Great questions. As a matter of fact, influenza has been, as we all know, has had pandemics in the past. 1918 is one that people particularly highlight due to the high death toll and large impact on civilization at the time. I think that the influenza is the most likely next pandemic, and that's why we have to particularly take care in getting our systems up and running. That's one of the benefits of also making seasonal vaccines, because we can prevent disease today while we prepare for the pandemic of tomorrow. One of the ways in which we're thinking of and working to prepare for those future pandemics and for seasonal vaccines is the use of this new technology that we're talking about, this self-amplifying mRNA vaccine. This is really a next generation version of the RNA technology. SA stands for self-amplifying mRNA. And like mRNA vaccines, the sa-mRNA helps protect against infectious diseases by giving instructions to the cell so that the body itself can make the antigen of interest. And by doing this, as the body makes this antigen, the body responds to it with its immune responses in order to recognize when it's infected in the future. And ultimately, this immune response is put into memory so that it can protect you in the future. But one of the benefits of the self-amplifying mRNA technology, as the name implies, is this can potentially enable you to put in a smaller dose of RNA and yet get either the same or even better responses than RNA, because initially there's a amplification of the RNA within the cell, and then the cell makes the protein of interest. So these two rounds enable a much lower dose.
Well, talk about the data that was published in Molecular Therapy, Methods and Clinical Development, some of the implications and why these implications are important.
Well, we're looking at the self-amplifying mRNA technology very broadly, thinking of it as a real tool to how we can address infectious diseases. In the study, we looked at targeting two different types of viruses, H5N1, which is a type of influenza that could potentially lead to a pandemic, and H1N1, which is something that circulates currently, and that's in our current vaccines. And in our case, we recognize that there are two main antigens on the surface of influenza virus. One is called hemagglutinin, or HA. That's the H in the H1N1. And this is where most vaccines today are targeted. However, there's this other antigen, which is also on the surface and actually doesn't change as much, which means it's more constant over time, and that's called neuraminidase, or NA. And so these are both required for the virus to infect cells and ultimately get out of cells and infect neighboring cells. And so what we did was we developed a vaccine containing just the genes of these HA and NA, these hemagglutinin and neuraminidase, and we looked to see, could we raise a robust response in animal models? Mm-hmm. We went all the way to the point of actually looking at these in a challenge model, which is an interesting model in animals where you first protect them with your vaccine and then you challenge them with the virus. And what we discovered was that not only does the hemagglutinin protect the animals that we were using in this model very well against influenza, but when you added the neuraminidase, this extra component, which is in some vaccines but not all vaccines, actually they developed an even stronger response, such that we could use tenfold less vaccine and yet get the same amount of protection. So really it demonstrated that using this methodology, this new technology, sa-mRNA, using two antigens, hemagglutinin, the one that we normally focused on, and neuraminidase, a new one, which is in some vaccines, but that we haven't really focused on historically, you can get a really robust response, at least in an animal model.
What's next in development?
Well, as we continue to work on this technology, there's a variety of ways in which we aim to continue to improve it. As I mentioned, the neuraminidase is highly conserved among influenza viruses. And so one of the benefits of that is it could lead to broader responses, which means that when you have broader responses, even if the virus changes during the season, you may actually get greater protection. So this is part of the design of the vaccine, is to try to do that. But soon we're moving this vaccine, which we've now tested in preclinical models, into clinical trials in this upcoming calendar year.
Well, is there anything that you'd like to add?
Well, I definitely want to mention that, of course, we're focusing on flu, but we're working more broadly against other targets as well. There are still vaccine preventable diseases out there to which there are yet to be vaccines. And that's very important as we continue to not only develop the platform, but continue to focus on human health, not just in a seasonal or endemic context, which is what one would say for influenza, but also in other contexts as well when we think about pandemics. To that end, we've just finished building a new state-of-the-art R&D facility in Waltham, Massachusetts. This is a 140,000 square foot facility to serve as a central hub for R&D for our company, focusing on vaccines, but also developing new technologies that can aid with vaccines or potentially even broader. So we are excited that we have that new site, and we're quite interested in further developing collaborations across industry and academia in order to really support the development of these new technologies and new vaccines. We're hoping that as this site comes on board, we will bring together a variety of different technical functions to integrate and collaborate quite broadly. You may see more about this, and in fact, if you visit CSL Seqirus newsroom, you can read our press release about this site as well as the manuscript.
Ethan, I appreciate your time this morning. Thank you so much for joining us here on Health Professional Radio. It's been a pleasure speaking with you.
Thank you, Neil. Very much appreciate it.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Ethan Settembre. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at Anchor, Spotify, and be sure and subscribe to our YouTube channel at youtube.com, Health Professional Radio.